So I am a medical geneticist, and I went to medical school. I then did a couple of years of internal medicine, and then fate intervened. And you always wonder about things happening, and it turns out that I was in the last class of physicians that was going to be drafted for the Vietnam War. And that was not a thrilling prospect, and I was looking around. I was all set to go to an Indian reservation, and then a position opened up at the NIH to do in a research lab. So I ended up going to the NIH. I worked there for 3 years, and we worked on collagen molecules and how they worked and all of that sort of stuff. But at the same time, there were 3 people who came down from Hopkins, each one of whom brought samples from people with a different kind of condition. One was with vascular EDS, the second was with osteogenesis imperfecta, or brittle bone disease, and the third was with a rare form, another rare form of Ehlers-Danlos syndrome. And over the next year and a half, while I was there, all of them were solved. So this was the very first clear indication that there were abnormalities in making collagens that gave rise to these conditions. And the paper that arose from that was about vascular EDS, of which I wasn't part, was alterations in type 3 collagen cause Ehlers-Danlos syndrome type 4. Very confusing, because you never knew whether it was type 4 collagen and type 3 Ehlers-Danlos syndrome, or, I mean, it was really confusing about that, and people have always had problems with it. But it was really the beginning of it, and then I went from there to do genetics and training at the University of Washington in Seattle, and then sort of sharpened my elbows and stayed, and I've been there for a long time ever since. And what we did was really to start, what I was trying to do was to look at how does one single building block of DNA, so DNA has 4 letters that make it up, A, C, G, and T. And why does the changing of one G to something else there, out of 3 billion building blocks of our entire genome, why does that give us, if it happens in the type 3 collagen gene called 3A1, so that's the gene, that's the name of the gene. So this is collagen type 3, and the protein chain that's made is alpha 1. Why does that one change, one lousy little change, make a difference, and make the difference between somebody who has a normal vasculature and a normal GI tract, and somebody who has vascular EDS? Vascular EDS is really a misnomer, because it says that the only thing that happens to people is that it happens to the vasculature. And in reality, many of you know that there are other kinds of things that can happen as well. So we've spent the last few decades trying to figure this out. And one of the things that happened is that we created a diagnostic testing service. And so we were able to accumulate information on people. And up until this point, we've now tested and identified about 1,500 people around the country who have vascular EDS. So it's that's a fair number of people. And it's been very hard to estimate. So how common is this condition in the population? Right? So we can estimate on the basis of the number of people we've studied. And then I've talked to people who run other diagnostic labs, and we think that there are not quite as many as the total number that we've studied in aggregate among all the other diagnostic labs that have been at work at it. So that would mean that there are at least 3,000 people in the country who have been identified. So 3,000 people out of 300 million people. We actually have a few more than 300 million people. So that's a frequency in the population of about 1 in 100,000. We think it's more than that, because we know that there are classes of mutations that we're not seeing. So, for example, most of the mutations that we see end up producing an abnormal protein. But there's a small group of people who have a change in one of these genes that means that one of the genes basically goes to sleep and turns off. And so they make half the normal amount of the protein, but what they make is normal. And they represent about 3 to 4% of all the people that we study. When we look at another gene called osteogenesis imperfecta in the call one A1, it's a different gene, they represent about 40% of the people. So we know we're missing that whole group. And then when we look at the kinds of things that happens, the kinds of changes that occur, we know that there are classes of changes that we should be seeing that we don't. Now, how many of you have had genetic testing? Okay. How many of you have read the report? How many of you understand what the report says? 1, 2. Okay. Okay. So, and what did your report say? Do you remember?
Basically, there was a misspelling in the 3A1.
Misspelling. Okay.
So, but he has a good set,
Mm-hmm.
and he had a bad set.
Okay.
And the bad set is what causes the lack of the protein.
The protein being. Okay. And do you remember what?
Yeah, you helped me. What you said is my son had a mutation in one of the starting codons for one of the exons.
Uh-huh.
And so it's a one base pair mutation, and his DNA in the trimer,
Uh-huh.
it's shorter than.
It's shorter than the other one. Okay. All right.
I have it right here.
Right. Right. So the hard thing is that, you know, what does a report say and what does it mean and why is it different? So, and I'm going to talk about that because it's part of what Shane was talking about a little bit, but it also is a way of thinking about if we're going to introduce genetic changes, what are the kinds of alternatives that we could do? How could we treat people from a genetic perspective and do things? So, your report has 2 different parts to it. One is something that starts with a C and then a period. And then it's got a number, so 791. And then it's got a capital letter, a G, to another capital letter. So this symbol there means that G. So remember, DNA is made up of A's, C's, G's, and T's. And they're about an equal number in the entire genome. And they don't make any sense by themselves. What our body does is to take these in groups of 3, and it tells the cell how to make an amino acid, what amino acid to put in place. So this says to put serine in that place, which is an amino acid. There are 20 amino acids, and if you take 4 of these things and group them in groups of 3, you get 64 alternatives, 64 possibilities. And so some amino acids have more than one, what's called a codon, or a set of 3 instructions for making an amino acid, and some have, or one of them has only 1. So there are a lot of different changes that can occur, and usually the important ones are the first 2, and if those change, then you get something else. And you usually change only 1 at a time to get the change. So this tells you, this is called coding sequence. That's what the C stands for. And this is the copy, basically, of the gene that's going on. And then there's another number, which is P, and that starts with something, say, called gly, which is the shorthand for glycine. Glycine is the most common amino acid in these proteins. And then there's a number, and then there's another number, another 3 letters, ARG, which is arginine, it stands for. And it means that in one copy of the gene, at this particular point here, glycine has been changed to an arginine. And that substitution means that the collagen doesn't work well. So these are, if you look at a glycine codon, there are 8 different possibilities that can occur in making a substitution. Now, the other thing that is important to know is that if we look at a gene, you would think, okay, a gene starts here and it has all the information for making a protein that's in it right there. And it uses the full length. And the problem is that these genes are broken up into pieces. So there's some stuff for making protein there, there's some there, there's some there, there's some there, there's some there. And these are called exons. And the stuff in between is called an intron. So exon means expressed, And intron means intervening. It means it's in between the stuff that's making protein. So in order to make this work, the cell has to do something very clever. It makes a copy of the DNA in an RNA, which is a very similar molecule, but then it combines them. So it does that. It cuts them out. And so in the end, you get this information all put together, and then the cell can copy that or use that as a template for making a protein. So, and what we heard was that sometimes there's a mistake in doing this. And it basically, there's information, there's DNA that's in here, and it says cut here and join there. Very simple instructions. And all it takes is one lousy change in one of the sequence elements that's here or here, that it says, I don't recognize where I am. And so what can happen is that instead of including that, it just cuts it out entirely. And so you end up missing a small chunk of the building blocks of protein, of the proteins. And that's what happens. And that's one of the fairly common kinds of things that can happen. So why is this important, and what difference does it make? Well, for a long time, we really weren't sure what could happen. And we study people. Remember, we're a diagnostic laboratory. That's how we see people. And so we tend to see people or have information about them once, except for the people that we follow locally. And we've got about, I don't know, 60 or 70 people locally that we follow. Remember, Seattle is a small town. You could fit several Seattles in Houston, let me tell you. In fact, you could fit several states of Washington in Houston. But what we did, we wanted to know from this information is if we looked at the ages at which we could identify people, or identify particularly the age to which people survived, what could we do? So if you look at the US population, this is basically what the curve looks like. That is that most people do pretty well up until they're in there sometime around the mid-60s or so. And then people begin to shuffle off this mortal coil, as we put it politely, right? And that if you look here at this point, this is the point at which this is the age in years here, this way. And this is the percent of people that have lived that long. So about when we call life expectancy, it's a measure of about by what point have half of the people survived. And it's in their 60s and 70s here. So if we look at some group of people with vascular Ehlers-Danlos syndrome, people do pretty well up until here, and then it begins to fall off like that. And the median age here, that is the point at which there are half people who have survived that long and half of the people who have survived longer, is about 50. It used to be thought that it was in the 30s, but it really is much longer than that. And so one of the questions that you were asking is, if you get to this age, what's the story? Well, if you start a life table there, you really start back up here, and it's the fact that you have gotten to be 50 doesn't mean it's all over. It means you really sort of start again, and you have a different life table, and we don't know exactly how that works. But this is for the aggregate group of people with vascular EDS. And then we can say, well, we've looked and we recognize that, remember, we had these people who made half the normal amount, but everything they made was normal. Remember that? So if we look at them, they're actually out here. And we look at those families, they're very interesting in the sense that there is always somebody that brings those families to attention. I'll come back in just a second. Let me finish the thought, otherwise it will vanish along with everything else. That there's somebody in those families that bring them to attention. But when we go back and look at those families, there are people in their 60s, 70s, and 80s who have never had an event. Never. And they will die because of cancer, they get hit by a truck, the usual kinds of things that happen. You get a little unsteady as you get older, and so you're subject to all kinds of things, falling off a mountain because you've been climbing up it. I mean, all kinds of things like that can happen, just as they would for anybody else. And this group of people looks quite different. So that tells you something. That says if we could take that copy of the gene, remember, we have two copies, one that we got from our father, it's called the paternal allele, one from our mother, and one of these is altered for people who have the more classic kind, the more common kind, which is that kind where they make something that's an abnormal protein. If we could take that one and turn it off, we would turn people into this group. So that's one of the ways of thinking about how to do that. And there are beginning to be ways to think about doing that. It's a problem in terms of delivery, how do we get it to go where it's supposed to go, and things like that. But that would be one of the possibilities. If we begin to look here at the people who are not in this group, then what we see is that, when we get enough people, we can divide them up. So if you think about amino acids, there are big ones and there are little ones. If I could spell them correctly, I can't see them from this side. So big amino acids, and they include arginine, valine, aspartic acid, and glutamic acid. And then the little ones include serine, cysteine, and alanine. They're all, they're pet names for these things. So those are the abbreviations. You don't need to know them. There's not going to be a test. All you need to know is big and little, okay? And then you can go back to your reports and you can decide, do I have a big one, do I have a little one, okay? Or you can ask us and we can tell you whether they're big or little. So if you look at the people who have big substitutions, one lousy amino acid that's bigger than the usual glycine. So these are all glycine, which is the smallest amino acid, to either this or something in this class. These guys interfere with the structure of the protein more than the little ones. And so they generally have a slightly more, in aggregate, more difficult time than the average. And the ones with the smaller ones are out over here. So that's another thing. Could we do that? Could we simply change a big one to a small one? Well, that's a little more difficult. It's a little easier to turn one copy of the gene off. And then there are the other ones. So there's another class, which is, in general, remember we have this DNA over here, and it's got to go from having things separated to putting them all together, and this process is called splicing. So it's like taking a wire and cutting a piece out and putting it back together and doing that sort of stuff. These ones tend to be on this side because they act like a bigger piece of change in the gene. They act more like the big substitutions or the big amino acids that are there. So these are the kinds of things that we would think about in terms of treatment. So could we simply say, okay, you got this from your father. He's always the bad guy. Or you got it from your sweet mother, who's always the good person in here. Or it came out of the blue. So how many of you are the first person in your family, or your kid is the first person in the family that knows about it? Okay. Well, how did you get chosen? What was it that happened? Did your mother fall down the stairs? Was she drinking too much? Was your father in some place where he was exposed to things that were going to make DNA change and be different? I mean, these are all the kind. Did the telephone ring during that precious time when things were going to happen? And that disruption really, really made a difference. What causes that to happen? Well, think about it. every time a cell divides, a cell has 2 copies of every gene. There are 3 billion building blocks that you get from your mom. There are 3 billion building blocks that you get from your dad. It's got to copy each one of those in order to distribute it to the next generation of cell. So that's 6 billion copying events that have to occur, and they should occur perfectly. So you can think about it a little bit like a Xerox machine. So you put a piece of paper in and you copy it and you get it. It's a little fuzzy around the edges, but it's, pretty good. You take that copy and copy it some more. It's pretty good, but it's a little fuzzy around the edges. It's a little fuzzier, and do that. And you realize if you don't have a repair mechanism for fixing things when you copy, then pretty soon, you're going to be putting a white piece of paper in the Xerox machine. You get a white piece of paper back. If you're lucky, actually, you get a whole bunch of little black dots on there. But so we have this tremendous system of DNA repair when we copy. Our copying machinery makes mistakes. And that's part of the adaptation that we have in the world. That, our genome is always trying to find a way to make things do a little bit better. Sometimes it doesn't. It makes a mistake. One of the mistakes is a single copy out of the 6 billion is, has a single change in it and does it. So we could try and fix the copying machinery. That doesn't work. And in fact, if you look at every single cell in your body, if you're able to take every cell is genetically different from every other cell, because it preserves one or more changes every time a cell divides. And we accumulate 2 to 3 changes every time a cell divides. If that cell that's dividing happens to be in the germline, that is, in guys it happens to be in cells that are making sperm, or in women that are making eggs, then that change can be transmitted and can happen and can be transmitted for the first time. So some people happen. And if we go back and if we look, in almost every family in which there is somebody with vascular EDS, we can identify the first time in the family that it occurred. We have very few families where it goes back more than 4 generations. So we can almost always find them. Now these guys would be the exceptions, the ones that have half normal making it, half the normal amount, but the structure is normal. Those families we can see are quite long-lived, and we can see many people, but we have to study them and go back, and we are often surprised at recognizing what happens. So this gives us some idea. So Shane was saying, okay, how do we begin to look at kids, and how can we begin to get an idea? So what we need to be able to do is say, all right, here we have an adult population that's been sampled, and we can see who's in it, and we can predict from that. We can say, okay, so this group of people are ones where, as a class, now recognize this is a class, and if you look, these curves always come out here. So there are always people out down here at the bottom who are quite long-lived. And so this is not really good in terms of saying, I know what you got. I know the date. I'll write the date on a piece of paper and you're done, right? Okay. But that's not how it works, because we're really looking at averages. We're looking at populations, and we're looking at broad expectations, but they're very bad at predicting the absolute thing that happens to an individual. But we treat people, either because there's something exactly different about them that we know we can find something that exactly will fit that and make it better, or we can say we have a class of people who share the same thing, and we know that on average they're going to be in this group. So that's one of the things, and it certainly will be one of the ways as we go forward. So for example, in drug trials, we want to be sure that the kinds of mutations that are represented in the population is exactly the same in people who get different treatments. And we want to have enough people so that those classes are equally represented and we can compare those. So you could say, well, okay, we're going to treat people with celiprolol or labetalol or guantanamolol or any other kind of drug that we might want to make. And we want to make sure that the populations are the same and that we get the outcomes and we have enough people in the trial that we can measure them. And we can say, there really are subgroups in here and that the subgroups respond differently. And we want to be able to know that a lot. So can we realistically do this? Can we convert one of these to one of those? Well, we can do it in a dish, a Petri dish. But you're much more complicated than the Petri dish. Thank goodness, I mean, can you imagine having a conversation with a Petri dish? They don't respond very well. They just don't talk, You say, we've got to talk. The three worst words in the English language. We have to talk.
And if it did, you'd have a heart attack.
And if it did, I would have a heart attack, but it would be a willing one, let me tell you. If they could tell us what happened, it would be great. So theoretically, what we could do is to say, all right, we could say, all right, let's look for the thing that makes this not, functional. Well, that means that we would have to study everybody, and we would have to know the precise thing, and we would have to target that. But let's say there's a common event or a common variation in the gene that's about 50% of the people have one variant and 50% of the people have the other one. Or 50% of the copies of that. On average, this kind of mutation that turns the gene off will occur, or even these guys, on average it will occur on the background of this common variant or on the background of this common variant. So if we simply devise a way of saying, all right, let's turn all the copies with this one off and leave the one that doesn't have that, and the one that doesn't have it also doesn't have the deleterious, the harmful change in the gene, then we would have somebody who goes from being here to being here, if we could do that. You can do it in a dish with a certain degree of certainty. Not 100%, but we don't need 100%. We just need a lot. Can we do it in people? Well, think about the tissues. Think about where we have to target things. We have to get blood vessels, and we want to get the bowel, the GI tract. Those are the two most common places where things occur. And we need to get the cells that make the lining, the proteins in the lining, which is where the type 3 collagen goes. It's a difficult task, and we don't yet have good ways of getting it to hit the cells that do it. We could certainly, so we can have, people are now making animal models for this, and it at least has the substrate or the class of animals that would be a way to try it and see whether or not it works. So genetic treatments of that kind. So you read about CRISPR, and what CRISPR does would be to go in and try and alter the specific alteration that is in one copy of the gene here. And it can do it in a cell. There's some off-target effect, and you don't know how big, how often you'll hit something else and have something that you don't want to have happen. But that's one of the possibilities that could happen. And these technologies are evolving. They're getting better. They're getting more specific. But still, for somebody, if you have something that's in your liver and the liver cells are making something that is not right, you can get something into the liver cells, they'll take it up, and you can fix them pretty well. But if you need to do it in the entire aorta, and you need to do it in all the vessels that come off, and you need to do it in the full length of the colon, it's much more difficult to get that to target. And I think that one of the things that will happen over time is, you get the flu, you feel crummy everywhere because that virus gets you everywhere and does things. Maybe we can use things like flu viruses to infect the cells and to do it in a way that you feel crummy for a little bit, but you wake up and suddenly you have arteries that are in great shape. I mean, that would be the kind of hope that we have. Is it realistic? Well, I don't think in the next 2 to 3 years, but maybe as we go a decade out or something like that, we'll be able to transform the way that we have of looking at these kinds of treatments. what we have still coming down the road, I think, is exactly what Shane was talking about, and that is small molecule treatment. So what would you do? Well, what we know is, if you turn something off like this, could you say, oh, let's boost the other one. Let's get the other one working just a little bit harder so we're making more from that one. Or can we selectively turn off the ones that are making an abnormal copy of the gene or an abnormal protein? Well, maybe we can. Or maybe we can say, okay, these are not making things get together properly. We don't get molecules that have the right shape. Can we reshape them and do that sort of stuff? And there's a lot of work. So cystic fibrosis, for example, is a condition where exactly that approach has been taken to try and get these proteins to fold correctly so that they go where they're supposed to go. Can we do that with these? Maybe. Maybe we can. So these are the kinds of things, or the kinds of genetic approaches, so genetic approaches are one, but we have other approaches. And I think we should never give up on the idea that small molecule treatments, whether they're treatments with drugs that are antihypertensives, and is it the lowering of the blood pressure or maintaining the blood pressure low by itself, which is what Shireen thinks is really the question, is the thing to target and the way to get to where we want to go. And I think we need to be much better about doing that. So how many people do we need to do that? Well, we need a fair number. How many people do we have? We don't know. As I said, we think that we can account for about 1 in 100,000 people in this country who have this. We know that that's an underestimate, but how big is the estimate? So we can take advantage of looking at studies where a lot of people have had their genomes sequenced and ask how frequently are mutations in there. And if we take those at face value, we think that the frequency may be as high as 1 in 15,000 people in the country, which makes this not a really rare disorder. It makes it much more common than we think. They're just not coming to attention. So it's like all these people out here, they don't come to medical attention. We don't see them because they're surviving, they're doing just fine. All the people that are down here in the curve who are getting into problems in their 50s or in their late 60s, they're not being counted as having this. We think of vascular Ehlers-Danlos syndrome as a condition that gets you when you're young. Well, it hangs around for a while, and it can also get you while you're old. But if something happens out here, it's rarely thought about as the cause of what's happening. So somebody that has a dissection when they're 65, they're just somebody that has a dissection. Vascular Ehlers-Danlos syndrome is almost never in the differential diagnosis, but we need to be looking much more at people like that. So that's where we stand. That's where we are. Remember, reading the genetics things is important. You need to know what the vocabulary means. And it is like reading a foreign language, but once you've learned the very basics of the foreign language, you go a long way with it. And you don't have to learn very much. The vocabulary is not complicated. It's only vocabulary. There's not a single difficult idea in genetics. Nothing is hard in there. It's the simplest science of all. And basically, if you can count to 4, A, C, G, and T, and if you can count to 20 amino acids, and if you can count to 2, father and mother, you're home free. You've got the whole idea there in your head about how genetics works. And it really is, and it makes it easy. And it isn't hard. Any of you who have unaffected parents have more than one affected child? Okay. And did they figure out what you have? What kind of genetic change you have?
Milowitz did a testing in 2008.
And?
I don't know any more than that right now.
Okay, you don't know anymore. Does she?
Milowitz?
Yeah. Did she tell you what's
the...
the...
Tried to get me into a study.
Yeah?
And I freaked out back then.
Do you feel any better about it now? You will.
Well, she's had a bad episode. You just got in the hospital.
Yeah, yeah.
We
So,
lost him for a little while.
Uh-huh. But you came back. Great. That's terrific. So it may be helpful, because I would think that you might actually be in this group out here.
I can't hear you.
You might be in this group out. That's because I don't speak very loudly sometimes. You know, I realize it's a problem. Well, there you go. It's a two-sided story, right? It's the donor and the recipient, as we say. Anyway, it would be helpful in a sense to know what it is that the change is.
I have my testing paperwork at home.
Yeah. Okay. So go back
It was
and
done in Seattle. Testing was all done in Seattle.
we did it.
Yeah, you did it.
He had his first episode when he was age 40,
Uh-huh.
so he didn't have any other symptoms.
Yeah. Yeah.
So maybe the whole mutation.
Yeah. So if you talk to me, we can look you up. If there's internet here.
Yeah, I have internet.
You have internet here. I can't look it up on your phone, but I can look it up on my computer.
There's Wi-Fi here too.
Yeah, that's what it. I know. Anyway, so that's where we stand in terms of genetics, in terms of treatment. It's the most exciting area of medicine now. And if you didn't think that I thought that, then I clearly didn't get the message across. But it is also the future of medicine. And we really think that, you happen to have one gene that's involved, but the same principles are present for many people with all kinds of other things. And that the genetic factors and the variations that we have are going to be the clue to how we treat people and how we make diagnoses and what we do as we go down the road in medicine. And you need not to let people forget that. Okay, so questions? Yes. Okay. Okay.
But I was gonna say, as far as there are some genes that have more mutations than other genes,
Mm-hmm.
and I was wondering how common, like, is the COL3A1 gene? I think I got this all right. I don't know, but how often do you find mutations in it? Is it prone to having more mutations than like other genes, or is it pretty conserved, generally speaking?
Well, the gene, yeah, so the frequency with which we find mutations in genes depends on their size. That is, how big the proteins are that they make. It depends a little bit on the sequence of the protein. And it depends on how important it is to have the exact stuff that's there originally. So in the coding sequence in collagen, in these collagens, 2 out of every 9 nucleotides or DNA building blocks in one portion of the gene that encompasses about 2/3 of the gene is susceptible to mutation and will give rise to a clinical picture. So that's a very high density of mutations. When we look, so far, let's see, there are, in the database that's reported publicly, there are about 600 unique mutations. So, and we know that there are two mutations that are much more frequent than usual. One is a splicing mutation, which is in intron 24. And the other one is a glycine to serine at position 16 in the, which is actually position in the protein, is position 183 in the protein. So there are about 30 of those, and there are about 40 of the splicing mutations out of the more than 650 or so that we've identified. So they represent a few percent. Almost every other mutation is individual to a family, or there are just a couple of families that have it. So there's a lot of space, and there's still a lot of space to fill up in terms of other changes that occur. Okay. Yeah?
Is there anything specific to null or
Haploinsufficiency.
need to be aware of?
What kinds of things are you thinking about?
Just, I guess, we know that our family has that.
Mhm.
That's the mutation, but we
Yeah, yeah,
In addition, there's little information on families.
on what happens with them, yeah. So that's this group here. So haploinsufficiency, haplo means half, and insufficiency means insufficient. I don't have another good word for it, but so that you're missing the product of half. And so what it means is that the protein that's made is normal. You just make about half of it. So, as I said, so there are people who are here who really never have any complications at all. And that's very striking. And so I remember one of the families that we saw a few years ago, it was an 18-year-old girl that actually brought things to attention, and she had had a dissection in one of her cervical arteries and had a constant headache. We don't know whether that was acute or whether it had been there for a long time, which is actually, these are quite common in people with vascular EDS, and it happens. And she came with her dad and her mother and her brother, and her mother was convinced that she had it because she was the mother. And in fact, it turned out it was the dad, and he was an avid mountain climber and hiker, and his father had had bilateral knee replacements. He was 80. He'd had one heart attack when he was in his 70s. So what we know in general is that the people in this are susceptible to things happening, usually at a later age, and usually not the dramatic kinds of things. So on occasion, they've had aortic dissections, and they've been repaired and replaced without any major difficulties, because the tissues are actually pretty easy to work with and much more like the usual ones. What we don't know is, why does that one person in those families come to attention? You know, what happened? What was it about the 17-year-old that made her different and made us see her early? And that's how we were identifying the families. So we think that there are a couple of possibilities. One is that, you know, you have these two copies of the gene. Are they the same? I mean, let's say, you know, except for if we look at them before they've had mutations in them, are they the same? And the answer is probably not. That is, some are really oomphy, you know, pump out a lot. They tell the cells to make more than half. Others of them are kind of weak-kneed, and they say, you know, I'm going to be a little lazy. I'm not going to make quite as much. So if the other copy of the gene that you have is one that doesn't put out as much, then maybe you're having complications up here. And if you have a really oomphy copy on the other side, you don't get anything, or you're down here. So those are the kinds of things that we're interested in looking at. And we've tried to determine how can we tell the difference between copies of the genes. And we were able to look. There are variations along them which are just, you know, ordinary variations that people have. And we've been able to identify about between half a dozen and 10 different copies of the gene, and now trying to look to see whether, you know, whether there's a difference in what the product is from each of the genes. And one of the problems is that we have as cells to look at skin. Well, skin, they make the same molecules, we think. They make them from the same genes, but we don't know that what controls their oomphiness in skin is the same as what controls it in the artery. So, and it's not considered good form to biopsy arteries, unless they have surgery on that artery and we can get a piece of it. So we're a little bit limited in doing that, but those are the kinds of things that we would look at.
So am I understanding you correctly that there's variation, even within if you have haploinsufficiency, there's variation so much within the family that some come to attention?
Well, we find them because somebody comes to medical attention. And they come to medical attention because they have the same kinds of complications. It's enough to lead us to that diagnosis, or to lead somebody to the diagnosis, yeah. But when we've gone back and looked, you know, I don't think we've found anybody who is the first person in the family who has that class of mutation. So those families are out there. We're just not seeing them. So if you have a drug trial, for example, and somehow in one half the drug trial you've got 50% of the people who have haploinsufficient mutations, and the other one you have 10%, and it's the half that in the 50% that's being treated, that's going to be a great drug. because, people are, the rate of complication is going to be much less. So that's one of the issues about those trials. Yeah.
Is there anybody out there doing research on null mutation within vascular EDS?
Well, there is a mouse that has nulls, and they initially they didn't have anything. So that's one part. That's one way to do things. And then so there are the mice that have been looked at. Not much is being done with them at this point. We're very interested in the families that are like that, and would love to have more information about them and know what it is that's going on and what's happening. And did we do the testing in your family? Huh?
Sign us up.
Yeah. And did we do the testing in your family?
Well, I don't think you guys did, but we have the genetic test.
Yeah.
We had a whole exome sequencing, and we're participating in a few studies.
Uh-huh.
To my knowledge, it's not specific to
And did you have the whole exome sequencing because somebody was looking for something else? And
it was an incidental
it
finding.
was an incident. Have we talked?
You emailed with my
Yeah,
husband.
we
about a
emailed
year
it
ago.
about a year and a half ago.
We can talk offline.
Yeah.
I didn't mean to
Yeah,
cut the whole
yeah.
talk, but
There are two families that I know about where the COL3A1 mutations were found as incidental findings in exome. So I'm always interested in knowing more about it.
we'd love to talk.
Yeah, I'm here for the day. That's why we're here. I mean, that's the whole point. Yeah. Okay. Oh yeah.
So over the years, I've learned that there's three, like you find the severity of vascular EDS, there's three kinds. There's the splice, the lysine,
Mm.
and the null, which is I guess the null is the haplo.
Is the
It's the same
haploinsufficient?
thing, right?
Yeah.
Is it still the same thing?
Yeah.
Or have you
Pretty much.
found out maybe something new? that we don't
There
know?
are other classes, but they're small. So there are some where there's a little piece that's missing, just a little piece that's missing. There are some where the assembly of the chain that gets put together, but they behave either like they're in one class or the other. Yeah.
So if later on when I meet up with you personally, you can look up on your computer, because my daughter was
Mhm.
tested through your
Yeah.
organization. You can let me know. I know she's glycine,
Yeah.
but I don't know how much collagen
Okay. Yeah.
is defective.
Yeah.
And I want to
We
know
can look
that.
at that.
I always wanted to know that.
Yeah. Okay.
Okay, okay.
Actually, pretty much. Because my daughter's also splicing, so I was wondering if that falls in the category of like severe or more severe.
So how do we know where? So people will fit into a class, but by themselves may not represent that class very well. That is, there are people who have what looks like a relatively more severe kind of mutation, but they're out here on the curve and doing very well. So we don't, and that's because, if you think about the haploinsufficient families where, one copy is not working well, and there are people that don't do as well in that group. We have people, so one of the things we did was to say, okay, let's take all the people who have a specific mutation, and then let's look at what their survival is. And so if this is the age up here, what we see often is that we have, people who have died at, these different ages. And and yet they have exactly the same primary mutation. So remember, we have 20,000 other genes, and we have all these other kinds of things that modify us and all of these kinds of things. And that there's a lot of stuff out there that's going to make a difference in this. And that's what this represents. So that's why, if we see this person at the age of 2, this is the question that Shane was saying, what do we do? How do we make treatment different? Well, we can say, okay, here's the average of what happens, and we have another group where the average is up here in terms of survival. We can say we're going to take this group and treat them as if they are like this, and maybe this, but then when we look at this group, we'll see that we have the same kind of scatter that's like that. It's just that on average, when we look at what happens, their life expectancy is longer. So it's not very good for the individual. It's okay for the class of people. And we have some families. So for example, one of the families that we were following in Seattle, the first person that died was the grandfather. He died at 39. He was in a water skiing accident. He ruptured his spleen and he died. The second person that died, she died when she was 39. I forget what it was. The third person in the family died when she was 39. The fourth guy got to be 40, and he had a big blowout celebration because he got beyond the thing, and he died at 42. His daughter is now 60. So, you know, here we have a tight band in some people, but clearly something that's modifying it in other people in the family, and we can't tell who they are at the beginning. We'd love to know, and that's one of the things. So that's the other part of genetic treatment, is if we can figure out what genes are doing that, if we're figuring out what genes get you to here instead of to here, maybe we can take advantage of that and we can sell that product. You know, or we can buy enough of it for everybody, you know, to spread around and put it in the water. Yeah. There was somebody, let me come back to you in a minute.
I was going to ask now, if you did the testing, like say 25 years ago, would there be additional information that would be in the reports if you got retested today?
So was it a genetic test?
Yeah, you diagnosed her dad with the vascular Ehlers-Danlos.
So the report would look different because we make it easier to understand now. We have a little table that has that C and the P and that stuff in it.
That's what I was wondering, I didn't remember seeing.
Yeah, that was all written in text there. Or 25 years ago, we might have just studied the proteins and not studied the DNA. So, but if it said that there's a specific mutation, the mutation will not have changed over time. What we have now is more information about where that fits. So, but we don't, you know, so we usually in our reports we say that, you know, this kind of information can be used to stratify in terms of groups, to order, you know, what kinds of things might be expected, but it's not very good at the individual level, and that's how we've modified things before there.
Would you suggest retesting?
No,
It's been that
no.
long, that
If the DNA test was done, the DNA hasn't changed in 25 years. Well, not that DNA anyway.
Right.
No, it doesn't. So the DNA is a permanent, it's on your permanent record.
Okay. I just didn't know if there would be more information to be gathered.
No. There's more information in the sense that we have much more information about the natural history and how it's related to the kind of change. But that you get from, you know, reading some of the papers and things, or we send you a copy of the paper and we send you a little translation guide that says this is how to read it. You know, or we can, you know, we can tell you it's important.